Welcome back, everyone. Thank you for joining us for episode 2 of the Immunology for Everyone podcast by SKA. This is hosted by Professor Dr. Sunil K Arora, whose amazing 40-year stint into the field at the PGIMER Chandigarh was complemented with being head and member of many national and international immunological societies. His experience is enriched with many national and international collaborations and working with subject leaders from all medical specialties. It is an honor to hear and learn from him through these podcasts. Today, on the International Day of Immunology, 29th of April, he has chosen to give us a peek into the very foundation of the becoming of immunology to what it is for all of us today. The different factors that play a vital role in modulating the immune response are very much embedded within this intriguing background. I cannot wait to hear how his future podcast will be built out of these significant personalities and incidences that changed the science of human body forever. We appreciate your responses and expect comments, suggestions, subscriptions for even better future episodes. So, listeners, it is SKA for you.
Thank you very much. And hello, everyone. Welcome to this new episode and greetings on the International Day of Immunology. Nowadays, we see that whenever a child is born, the first thing done in the hospital is some vaccine shots given at birth, and some more are given within few days and weeks after birth, followed by some boosters at various intervals of time. The list of such vaccines has become exhaustive, and new ones are being added. We also see that all the mothers to be are also given shots against tetanus before they deliver their babies, because we know that this process will protect the newborn child from tetanus. And we now very well understand that this is because the antibodies formed in the mother's blood pass on to the newborn, and the phenomena which is called the passive transfer of immunity. In more recent times, we all were witness to the speed with which the highly active and efficacious vaccines could be developed for COVID-19, and how the mass vaccination against SARS-CoV-2 turned the pandemic around. We will take up the technical details of making a successful vaccine in one of the subsequent episodes of this podcast at a later stage. But we all now well understand that these vaccines are so important to provide protection against many infections and even cancer. For example, a vaccine against human papillomavirus protects against cervical cancer in young women. So the point I want to make is that the vaccines have been instrumental in saving billions of lives. And improving the life expectancy. But this was not true a few hundred years ago, as most of the deaths and devastation used to occur because of deadly infections like smallpox, cholera, TB, leprosy, plagues, etc., etc. And those were the times when the science was not advanced enough to bring out such remedies so quickly. But we had some visionaries during those days whose critical observations and some bold experiments laid the foundation of modern day biology and immunology. I would like to dedicate today's podcast to those visionaries who, in real sense, were the originators of basic principles of modern immunology. So let's salute them today when we celebrate the International Day of Immunology. And in that pursuit, I would like to take you all to those older times, remember some of those stalwarts and their work. So if we try to read the history, we find mentions of such observations as early as 430 BC, when a Persian physician Abul Bakr al-Razi from Iran stated that if one survived a disease, the person thereafter become immune to any subsequent exposures from that infection. In my view, this he said when there was no idea about causative agent of the disease. Due to the lack of understanding, even the societies in the West would take the diseases as a punishment from God or a curse at the maximum. Somewhat similar beliefs were mentioned in writings of Eastern culture also, where such diseases were considered due to some noxious vapors. So this confusion was there because nobody knew then that some microorganisms or pathogens were the cause of these diseases. And also because they did not have the advanced tools like microscope to see what is there in the pus exudates of, for example, smallpox or chickenpox. But in spite of all that, a procedure had become well established in China by 1700 AD, which was called variolation. So this was derived from the name of the infecting agent, the variola virus. So the basic principle of variolation was to deliberately cause a mild infection with an unmodified pathogen. Although the risk of death from variolation was around 2 to 3%, yet the variolation was considerable improvement on the death rate for uncontrolled infection. So similarly, immunity to smallpox was also conferred by inserting the dried exudates of smallpox pustules into the nose. This technique for transfer of smallpox as a form of limited infection also traveled to West from China. And interestingly, even the royal family of England had their children inoculated against the disease in early 70s. But the practice being too risky aroused a lot of opposition by the general physicians. It was Edward Jenner, whose name must be very familiar with all of you, as one of the initiators of vaccination, made a very important observation in 1792 AD when he found that the maid working in his farm got some pustules on her hands, which was similar to the pustules on the teats of a cow. Jenner observed that this lady had a mild disease from cow and recovered, but never contracted the severe form of smallpox disease, even when she was exposed to people having smallpox around in the neighborhood. He thought there must be some phenomena in the pus of cow's disease which induced only milder disease in humans and eventually induce a protection from the severe form of smallpox. To test his hypothesis, he did a very bold experiment when he injected the scraping from healed pustules of cowpox on the hands of that maid to both the hands of a young boy in his farm. So he found the boy had a mild disease and fever, but recovered in few days. Now Jenner injected the pus from active lesions from a smallpox patient into the hands of that young boy again, but to his surprise and astonishment, the boy did not get the severe form of smallpox. This led Jenner to postulate that the pus in blisters that affected individuals by cowpox, which is a milder disease than smallpox, but much less virulent, protected them from smallpox subsequently. And now you imagine that Jenner made those statements when neither at that point of time he had any idea of causative agent of smallpox or so for that matter, the cowpox even, nor he had the idea of mechanism of such a protection, which was induced by cowpox for against smallpox. Yet he termed his procedure as vaccination. Edward Jenner used the phrase in 1798 for the long title of his inquiry into variolae vaccinae, known as the cowpox, in which he described the protective effect of cowpox against smallpox. About 90 years later, in the honor of Jenner, Louis Pasteur, a famous scientist in those days, proposed that the term vaccination, as proposed by Jenner, should be extended to all the new protective inoculations that those were being developed in the lab of Louis Pasteur. And you guys are well aware that the science of vaccine development and production is now termed as vaccinology, which is a tribute to that great scientist and visionary. Now, the idea that a pathogenic organism caused the disease was not fully accepted until certain technological advances and had occurred. Initially, the development of a microscope by Antony Leeuwenhoek, who was a Dutch microbiologist. He discovered microscope in 1760s, and the subsequent realization that some very small living objects existed in some of these pustules, which were not visible to the human eye, and that allowed the concept of germs in biology. However, whether these living objects were causative agent of the disease was not appreciated until Louis Pasteur, who was a French chemist. He developed his germ theory of disease in middle of 18th century. Though his original interests were in fermentation, in wine and beer, and he was the first to isolate organisms that cause the fermentation process, Pasteur's work eventually led him to the development of a procedure which is famously called as pasteurization. That is by heating as a means of halting the fermentation. While working with silkworm and anthrax subsequently, he demonstrated that the process of transmitting disease from infected animals to uninfected animals was similar to the method of transferring the fermentation process through an inoculum, which he was using in his lab every day. So I find it very motivating that how some chance findings and acute observations have led to great discoveries. An interesting anecdote describes that in 1878, Pasteur accidentally used a weakened chicken cholera culture to inoculate some chicken, which did not get the disease. And when he tried to repeatedly inoculate those chicken with the fresh culture, still the same chicken did not get the infection. But another set of chicken got sick with the same inoculum from same fresh culture. So he got excited and tried to understand what was happening. So he repeated the experiment again with weakened culture, which he now called as attenuated by heating, followed by a fresh culture. Then he made a conclusion that the weakened form of organisms, when inoculated prior to infection with a virulent form, protected the chicken from the virulent form of the disease. And you see, this is the crux of vaccinology that holds true even today. So because of this interesting observation, Pasteur went on to develop attenuated vaccines against rabies, anthrax, and saved so many millions of lives. Now, the Robert Koch, another German physician who was working in the similar times and similar ideas in late 80s and early 90s, but unaware of Pasteur's work at that time, Koch was the first to isolate the anthrax microbe and showed that it caused the disease. Then in 1882, Koch made a seminal discovery when he isolated the microbe that caused tuberculosis. And the microbe was Mycobacterium tuberculosis. And then he demonstrated that germ theory of disease applied to humans and animals in a similar manner. These Koch's postulates are still used to identify the infective organism, the Mycobacterium tuberculosis. So much of the basis of modern medicine, as well as the field of immunology, can be traced back to these few scientists. But two major questions still remain to be answered at that time. One was, how did the infection cause the degradation of tissue in a disease like TB? And the second question, very important, was, what is the mechanism of protection induced by these so-called vaccines? So the first question was addressed as it is available in the history, that is in 1881 by Emily Rowe and Alexander Yersin, when they isolated a soluble toxin from diphtheria cultures. Rowe was a French physician, and he was one of the closest collaborators of Louis Pasteur and a co-founder of Pasteur Institute also. And he was responsible for Institute's production of anti-diphtheria serum. This was the first effective therapy for this disease. He also investigated other diseases like cholera, chicken cholera, rabies, and tuberculosis, using similar, strategy. However, the development of protection against diphtheria by transfer of serum from animals who were infected with diphtheria into healthy animals was shown for first time by Emil Behring, along with Shibasa Buro Kitasato. Behring got his first Nobel Prize of Immunology for the serum therapy of diphtheria in 1901. Eventually, in 1930, these serum factors were recognized as carrying antibodies. And now we know that how antibodies are protecting against the infection agents. However, 30 years before these antibodies were finally isolated and identified, Paul Ehrlich and some of his colleagues theorized that a cell under threat grew additional side chains to bind the toxin, and that these additional side chains broke off to become the antibodies that are circulated through the body. So Ehrlich hypothesized that these proteins were specialized molecular structures with specific receptor sites that fit each pathogen like a lock and key. So in 1908, Paul Ehrlich received the Nobel Prize for medicine in the field of immunology. So that's the first realization that the body had a specific defense system was introduced. In addition, sometime later, Ehrlich realized that this powerful effector mechanism used in the host defense would, if turned against the host, would cause severe tissue damage, which he terms as horror autotoxicus. Although extremely valuable, his work still left a large gap in the understanding how the immune system fights a pathogenic challenge. So it was Elie Metnikoff, whose field was actually zoology and who studied the phagocytosis in the single cell organisms, first coined the idea in 1884 that specific cells could directly be involved with defending the body. So Metnikoff also postulated that even vertebrates could operate in a similar manner to remove the pathogens. No wonder we find such cells as a part of our immune system from very early primitive organisms till humans in the evolutionary tree. However, it was not until 1940s that his theories were accepted and the cell-mediated along with the antibody-based immune responses was recognized as one of the protective mechanisms. So guys, we see that the classification of the immune response and the science of immunology did not progress in a systematic and chronological order. Many interesting discoveries were made many years earlier, but their real impact was recognized much, much later. Yet, once scientists had a basic understanding of the cellular and humoral branches of the immune system, What remained was the identification of various components of this intricate system and the mechanisms of their interaction. So, this could not have been possible or accomplished without the concomitant development of molecular biology and genetics. So, guys, I will end this episode with a note that now we can start discussing the basic concepts of both arms of the immune system, the cellular as well as the humoral. But before that, let me give you some homework. I'm sure you all must have seen the complete blood count reports of yourself, any of your close relatives, or even patients, for some of you practicing doctors. And there you see the list and counts of cells, which are termed as leukocytes, monocytes, lymphocytes, neutrophils, platelets, RBCs, etc., etc. And you see that your doctor is using these percentages and counts to relate with your disease condition, and so may be called your health condition. So please go and brush up your knowledge about function of each of these cells as an immune cell. And I'm sure you will enjoy our upcoming episodes where we discuss the important roles of these cells in generating the immune responses, both cellular as well as humoral, against various infections or cancer. So, guys, till then, thanks and goodbye.